think I got that one under 1 second break. All right. So how do you identify the pathogen? Well, morphology, what does it look like? Gross morphology, what does it look like with the naked eye? What does it look like under a microscope? What about biology, the host range studies? I'll show you a picture of a host range study in a moment. That'll make it easier. Biochemistry and physiology, so you can look at the proteins and the other biochemical aspects of the organism and what it's doing to the plant in the disease response. That can be clues. Serology is what's called enzyme-linked immunosorbent assay as the most common type of serology. And you're using the idea of antibodies that will attach to a substance with extreme accuracy, a lock and key mechanism to identify a particular protein. And I will show you again an example of that, but it's the same concept as a pregnancy test. If a woman is pregnant, they generate a particular protein that is excreted in the urine, and so a pregnancy test will turn pink, or a plus sign, a minus sign, whatever it does. It's basically indicating if that protein was there, it will bind to this special antibody in the kit. And if it binds, it changes color. It's designed to do that. And you can do the same thing not only with proteins for a pregnancy test, you can do it for proteins in specific viruses or bacteria or something else. You can do a quick test very easily, inexpensively, in large volumes to determine if there is a presence of a particular pathogen. And the molecular biology, one of the popular, fast, easy, cost-effective ways is a DNA technique called polymerase chain reaction. And I'll show you some photos to make all of these clear. Here's some morphology. Here's a couple of bacterial plates, and one of them is responding differently. It's fluorescing under this light. The other one is not. That's a clue. That's a morphological clue. Here's some fungi, all three related fungi, but very different morphological patterns. Here you've got a solid growth of gray with all of the black structures around the perimeter. This one, the black structures are all scattered throughout. This one, the black structures are much, much larger and fewer in just a handful of spots around the edge of the plate. So these are very similar fungi genetically, but their morphology is extremely different. Major clue. Here's some other related fungi, and you can see the patterns of orange and black. Very, very different patterns of color that give you a clue. Now, I talked about host range studies, and this is a host range study in spinach. And in spinach, you can grow up special seeds, not so special, they're just seeds that have unique combinations of genes that make them resistant to certain pathotypes of downy mildew. So if I know, for example, that this first one is resistant to downy mildew types 1, 3, and 5, and this one's 1, 4, and 7, etc. So I know which genes are in each of these. I grow up lots of that seed. I keep a whole bunch of that seed of every one on hand. I grow up trays of it, and now I have an unknown downy mildew. I know it's downy mildew. I can't tell from the mildew spores which it is. I can't tell looking under a microscope, but I can spray downy mildew spores from my unknown sample on my plants, And I can say, well, it killed number 1, 2, 5, and 6, but it didn't kill 3 and 4. And you look up on your little key and you can say, oh, this must be downy mildew type 7, whatever. That is a host range study. Quick and easy to do for certain crops. I've used it many, many, many times in lettuce and spinach for downy mildews. Now, they're not the same downy mildew, they're different ones. Even though they're both called downy mildew, the downy mildew that attacks lettuce is different than the downy mildew that attacks spinach. So here's ELISA. What do? You take a plate. This actually represents a plastic plate that's maybe about yay big. There are different sizes, but you know, maybe 2.5, 3 inches by 4.5, 5 inches in size, with a bunch of small wells or small little dips that are like mini micro bowls, if you will. And each one of these little circles can hold maybe 2 drops of water. And the plate is coated in a special material. In this case, it is lettuce mosaic virus immunoglobulin G. And the lettuce mosaic virus immunoglobulin G will bind with the proteins if lettuce mosaic virus is present in the sample. And if the lettuce mosaic virus binds to this, there is a change in the structure that starts to reflect light differently. It moves it, it twists it in such a way that it changes color. And because of this change in the molecular structure, when it binds to lettuce mosaic virus, it changes color. Remember what I said just a moment ago about the pregnancy test kit? It's the same thing. When it binds, it causes a difference in its light reflectivity. It reflects a color. And in this case, it turns a color in the presence of lettuce mosaic virus. So I take some seeds that I want to test. Is there any lettuce mosaic virus in the seeds I harvested for my crop? So I grind the seeds up, I put them in a buffer, I add that to the plate, and I determine, does it have lettuce mosaic virus in the sample or not? So here's an actual LMV plate. Here's some positive controls to make sure the test went right. A negative control to make sure that the system wasn't contaminated. And then the plates. I can say, and I can't see the numbers or letters over here, but let's say this was ABC4. C4 is contaminated. So on my list of samples that I put in, I can say, uh-oh, that lettuce seed lot that corresponds to the plate C4 is a contaminated sample. You got to ditch that sample, throw it away. It's contaminated with disease. These are the tailgate tests I said you could do right at your truck tailgate. All you do is you grab one of these strips out of a little container, you grab a plastic pouch, you put a tiny bit of buffer or just a salt liquid solution into the pouch. You drop in an infected leaf that you're trying to say, what the heck is this? You drop the infected leaf in there and hold the bag with the buffer in it on the side of your truck. Bang it a few times with the side of your hand to kind of squish it up. And drop the test strip in there and wait the specified amount of time. And then on the side of the container where these test strips came from, there will be a code, or a key rather, to read off. A certain banding pattern means there's no virus. Another banding pattern, if you get the other banding pattern, it says, yes, there is virus in that sample. And so this is tomato spotted wilt virus, I think. Impatiens necrotic stunt virus, cucumber mosaic virus. Again, I'm pretty sure those are what these are, but I don't know. I didn't make this slide. Dr. Kawicki did. and there might be other things with the same acronym that I'm not familiar with, it would say very clearly on the container from which these test strips came. But very easy, very good way to do the testing. I also mentioned PCR, polymerase chain reaction, and I'm not going to go into this here other than to say this is sometimes called DNA fingerprinting. And it's just one of many techniques of DNA fingerprinting. And so you run the sample through a process of extracting the DNA. You try and isolate the DNA in such a way that you can run a test. You do special things to try to amplify only the DNA of the disease you're looking for, and based on the presence of the banding patterns, that's a clue. And you combine your physical evidence of the disease symptoms. What are you seeing in that plant? This plant has disease symptoms, this plant doesn't. This plant does, this plant doesn't. This plant doesn't. This plant doesn't. This plant does. So you have a list of your symptoms, and you have your DNA fingerprinting or your PCR test results. You put the two together and it gives you enough pieces of the puzzle to hopefully determine what's going on. Very accurate. So, controlling plant diseases. Let's mentally go back to the disease triangle, all right? There's three things. I need a susceptible host, I need a pathogen, and I need a suitable environment. So as a grower, I want to try to remove one of those legs of the triangle so I don't get disease. I want to do everything I can to minimize the chance of disease, and I use the disease triangle information as a brainstorming tool in order to prevent disease. So what can I do about the host? Plant non-hosts. Remember the earlier slide? You got a problem with tomatoes with the late blight? Plant peppers. Plant something else. Plant-resistant cultivars. I have a problem with downy mildew in my area. Plant a cultivar that's resistant to the pathotype of downy mildew that I have in my area. Just find something that's resistant if you can. Sometimes you can, sometimes it's not available. Use disease-free materials. So if you're doing strawberry transplants or you're grafting a new vineyard, you want to make sure that you are getting things from a reliable source, that the materials have been tested in a proper lab, possibly even using newer techniques of tissue culture rather than field-grown transplants and stuff like that. If possible, you're trying to make sure that whatever you buy doesn't come pre-infected. Make sure your seed is tested. By law, you have to test lettuce seed sold in California for commercial use. You have to test it for lettuce mosaic virus. It's the law. You have to test it for everything else? No. Avoid damage to the plant. Damage just makes it more susceptible, more likely. Use proper cultural practices, proper irrigation, proper fertilization, proper pruning, proper planting depths, everying. A plant that is not stressed due to growing it incorrectly is going to be happier, healthier, and less likely to come down with a disease. And timing. You want to plant to enhance the plant growth. You want the plant to be happy and healthy and unstressed. Maybe you've got something where there's a disease in early spring and you want to grow some lettuce, but there's an early spring disease that really is a problem in that block, in that field, or that microclimate. All right, just do a crop of baby leaf spinach and then plant lettuce a little after that. Once you get out of that window, just don't do it then. And those are just some of the ideas. And some of these things you could argue could maybe go into other categories, but you should be able to draw the disease triangle, know what the disease triangle looks like, what it is, and you should be able to give me an example or two, at least, of every single one of the 3 legs of the disease triangle. What can I do regarding a susceptible plant host? Plant something else, plant something resistant, use disease-free material, etc. All right? What about the next leg of the triangle, the pathogen? Use pesticides, fungicides. Now it says fungicides here, but that only is for fungus. Pesticides would include the broader category of nematicides for nematodes, virucides for viruses, bactericides for bacteria, whatever it is. Try biological agents. There are certain biological agents for certain pathogens that can reduce their ability to compete or survive in your plants. Exclude the pathogen using sanitation techniques, using quarantine practices. If you're in a greenhouse growing environment or something like that, sometimes you can exclude the pathogen. Sometimes you can't. And if you're out in the field or out in the forest, or exclusion might not be an option. But in some situations, exclusion is an option. And physically removing the pathogen. In some cases, let's say I've got a vineyard and I've got some sort of a fungal problem on one of my vines, cut off the bad part or take out the whole vine and burn it, take it to the dump, whatever, get rid of it. And with some pathogens, they don't spread that fast and they don't take over areas that well. And so by just physically pruning it out, removing it, digging it out, whatever it is, that can be enough. Sometimes that's a useless effort. With certain pathogens, it's not going to make any difference. By the time you see something to remove, it's too late. It's already spread around in other areas. And the third option, the third leg of the triangle, what about the environment? Again, you want to use proper planting methods, fertilizers, planting depth, transplanting. You don't want to J root or have a curved root under the ground, bent root when you're transplanting your grapes or your strawberries or something. Use proper irrigation and fertilizer, proper sanitation in your field. You don't want to pick the bad strawberries and just drop them in the furrow. In some areas, for some disease situation, maybe you can get away with that and just throw the bad strawberries in the furrow. Other times, you want to take it out of the field because strawberries sitting in the furrow is a fungal factory. And you want all those fungal spores in the furrow just a foot from your plants? Maybe not. Probably not. Again, in really dry environments in certain areas with certain kinds of pathogens, maybe you don't care. The cost of taking the strawberries out of the field isn't worth the effort. What about weeds around your field? Weeds can host pathogens. What about cleaning your tools? If I'm pruning a vineyard, what about pruning shears being cleaned between every row or every block or every vine? You know, it depends on how bad the disease is and how scared you are of it or how present it is in your area, how stringent you're going to be about cleaning tools. Managing weeds and managing vectors. Vectors are primarily insects that spread these diseases. Things like aphids are notorious for spreading viruses, but that's just one example. When I was growing lettuce seed and I knew if I had a lettuce mosaic positive test in one of my seeds, I would have to throw out the entire lot of seed. It's contaminated by thousands of dollars of seed, tens of thousands of dollars of seed down the drain. I was pretty fortunate. I think I only lost one batch of seed, not that big of a one, in all the years I did lettuce seed production. Why? Oh, I was ruthless with weeds, and I would talk to my neighbors. I would go upwind for literally a mile, and anything, anywhere, I would try to stake out where are the potential problems, get to know my neighbors, and make sure that they're handling their aphid populations. And anyway, so overall appropriate crop management. In general, an environment that plants are happy, you're less likely to get disease, but you also want to keep things down. And there's some here that I want to add. What about in the greenhouse? Greenhouses are changing the environment. What about using shade cloth? That's changing the environment. What about pruning properly for your canopy on your grapevines? I'm growing wine grapes. If I don't prune the canopy properly, my canopy is going to be full of too many leaves, and it's going to get humid and shady inside underneath that heavy, dense canopy. Humid and shaded sounds like a perfect environment for fungal growth, and it is. And so you want to make sure you prune properly in the dormant season, and you train and trellis your grapes properly during the growing season, and maybe even do some leaf pulling during the growing season to allow airflow through the canopy and a little bit of dappled sunlight to come in. That's changing the environment. Putting bird netting over certain crops. What does that do? It changes the ability of birds to get at your grapes. And if the birds can't get at the grapes, they can't damage the grapes. And if the grapes aren't damaged, there's not the environment of damaged grapes to allow fungal infections or bacterial infections to wipe out my grapes. So again, there's lots of things you could think of with controlling the environment. Using mulches. Anyway. So diseases of plants, certainly it happens in everything, vegetables, fruits, ornamentals, everything. It affects every part of the plant. And there's a variety of pathogens, and these are the three biggies. As I said earlier, nematodes would be kind of the fourth for most of the people that I know around here. Nematodes being very important as well, and to a lesser degree, the others on the list, the phytoplasmas, the higher plants, the algae. So here are some examples of some diseases. I'm not going to go through that, but clearly none of these plant parts are happy. Some more unhappy plants. Some types of disease symptoms can be extremely surprising. First time I saw this, and I can't remember what it is right now, but if you do diamond-shaped stem lesions and you search Google, you'll find it, because every time I see this, I go, what was that again? And I do diamond-shaped stem lesion, and I go, oh, that's what it was. Google is good for some things. This looks like you almost shot it with, rock salt out of a shotgun or something, some sort of a bacterial leaf spot, I assume, some sort of a mosaic virus, I assume. But again, this is Dr. Kawicki's presentation, and I don't remember exactly what all of these things are. So there are lots of issues. Current issues include the actual lists of pesticides and fungicides that are available to growers. The lists are reviewed constantly. Every single pesticide has a 5-year review process. And so every year something is coming up on the 5-year review, and some things get phased out, some things get newly approved and licensed for use in California. And so there's a constant rotation and issues of trying to see what's being phased out or eliminated, and what are new things on the horizon that we might use. Conventional versus organic, I think they feed off of each other very well. Organic is pushing the industry in certain ways that are important, but it certainly doesn't have all the answers. Biotechnology and GMOs. In the vegetable crops, GMOs is a big deal. I won't get into that here other than to say that most people don't want GMOs in their fresh vegetables for some reason. Actually, most people don't care. Enough people care about their fresh vegetables with GMOs that growers aren't willing to grow them in most fresh vegetables, with some exceptions. There's lots of GMO corn on the market, and some other things, but I don't know, there's something about it in your tomato and lettuce that is different than in your corn. Go figure. But there's still a lot of issues. This is the same list for years, and it doesn't change. International trade and plant quarantines, that's a constant battle that's going on. At any given day, you would be amazed at the number of things that our ag commissioners have to deal with, and the California Department of Food and Agriculture, CDFA, in terms of what things can be exported from every county in California. There's always a long list of things that, nope, you can't take this crop from this county and move it to wherever. And then there's a whole list on the other side, where can't you send it? And then there's the import side. What can you bring in? Well, we'll take Canadian wheat, but we won't take you know, Chilean broccoli, or whatever, because of some disease that's going on, some pathogen. And food safety, always, always, always a huge deal. Here's a book written by Dr. Kawicki. You could look it up. It's variously available. This is the European version. You can tell it's the European version by the spelling of the word color. But the concept of vegetable diseases doesn't matter where you are. The concepts are all basically the same. This is a fabulous book. It's not cheap, but you could certainly see if you can get your hands on a copy if you're interested in pathology. I think it's a fabulous, fabulous book. And that's it for plant pathology.